Today we will learn thyroid hormone. In the first part of this lecture, we will learn how thyroid hormone is synthesized. But before that, we will learn a very basic detail regarding this thyroid hormone. All of you know that from hypothalamus, one hormone is secreted. That hormone triggers some special cells of pituitary, and from pituitary, as a result, another hormone is secreted. That hormone finally comes and stimulates the thyroid cells or thyrocytes. So from thyroid, the third hormone that is released. Now hormone number 1 is called TRH, thyroid releasing hormone. Hormone number 2 is called TSH, and hormone number 3 is T3, T4, the thyroid hormones. So precisely, in thyroid profile, though we are writing in our prescription T3, T4, TSH, TSH is not a thyroid hormone at all. It is not released from thyroid gland or thyroid cells. TSH, that is thyroid stimulating hormone, and it is released from pituitary. It has receptor on thyroid cell, but true cells it is not at all thyroid hormone. Thyroid hormones are T3 and T4, and sometimes reverse T3. We'll discuss in detail. So first we'll have to learn uh, how thyroid cell look like. Now thyroid cell are arranged in follicular pattern. If we want to see one kind of this follicle, so this is one follicle in three-dimensional structure. This is one follicle, and it has central hollow space. That hollow space is filled with colloid sometimes, and at the periphery, the blood circulations are there. Okay, so every thyroid cell, all the thyrocytes or thyroid cells, they are present in follicular arrangement. They has one basal surface, that means where the blood vessels are present, and one central luminal surface, but no blood vessels are present in the central part. So in next diagram, we will learn in detail how thyroid hormone is synthesized. Suppose this is one such cell, and all these thyroid cells containing nucleus. Now every thyroid cell on its basal surface, they contains a TSH receptor. This kind of G protein coupled 7-passed serpentine receptor or GPCR type of receptor. So TSH comes and binds with this receptor, and inside the G protein is there. I am not discussing the detail of the signal transduction cascade, but finally what happens? The nucleus is stimulated, and when the nucleus is stimulated, from DNA transcription process occur, and after transcription translation process occur, so some new proteins are made. One such new protein that is called thyroglobulin. Students sometimes confuse with this thyroglobulin with thyroid binding globulin. Those two are different. Thyroglobulin is a protein or peptide that is synthesized within the thyroid cell, whereas thyroid binding globulin is a transporter protein that is synthesized from liver, secreted from liver, and present in the systemic circulation. Okay. Now, before learning this thyroid molecule, we have to learn first how iodine is trapped within the cell. The source of iodine is dietary iodine from our vegetables as well as our table salt. So, iodine, with the help of one transporter, iodine will enter within the cell, and once iodine enters within the cell, this iodine will not go outside. That is why this process is called iodine trapping. Now, iodine in the form of iodide, negative charged iodide, is entered within the thyrocyte or thyroid cell. Now, this is a symport where along with iodine the sodium molecule is there. So sodium helps in transport of iodine. That is why togetherly this transporter is called NIS, sodium iodide symporter. Now iodine is within the thyrocyte. This iodine will go to the opposite side. Here in the opposite side, one specific transporter is there that is called pendrin molecule, and through the pendrin, now the iodide molecule comes outside. Now, here in this central hollow space, one ectoenzyme is present. That enzyme converts converts the iodide into I plus ion. This reaction is peroxidation. This reaction is peroxidation. So the name of the enzyme hanging outside, that is called TPO or thyroid peroxidase. Why thyroid peroxidase? Because this iodine finally will help in making the thyroid hormone. That is why, though the name is not appropriate here, but we should remember that it is TPO, but thyroid peroxidase. So I minus to I plus reaction occurs. Now few I minus and I plus together they will combine to form I2. So iodine trapping occurs first, then there is peroxidation of this iodine molecule. Now, in the meantime, the thyroglobulin, which is a polypeptide, that thyroglobulin molecule is rich in tyrosine amino acid. It also has other amino acid, but it is rich in tyrosine. This capital T denotes the tyrosine. Now this thyroglobulin molecule, they will also come here. This is the thyroglobulin molecule. And here iodine will attach with this tyrosine residue. Iodine will attach in this tyrosine residue. I am using another slide for proper understanding. Suppose this is one thyroglobulin molecule and iodine comes and iodine binds with this tyrosine amino acid or tyrosine residue present in the thyroglobulin molecule. So we can easily appreciate that for the first tyrosine, though the number is random here, for the first tyrosine, two iodine molecules are attached. Then here also for the second tyrosine molecule, two iodine, for the third two iodine, for the fourth one iodine, and for the fifth one iodine is attached. Now the process through which iodine is attached with an organic compound, that thyroglobulin, this process is called organification. Organification. So the steps are: first there is trapping, then there is peroxidation, then there is organification. So iodines inorganic, now they are part of organic compound. That is why the process is called organification. After organification, there is fusion of two adjacent tyrosine molecule, that is called coupling. What happens actually? Suppose this is one tyrosine molecule, this is another tyrosine molecule, this is one tyrosine molecule, this is another tyrosine molecule. All the tyrosine molecule are part of this thyroglobulin chain. Now, for the first one, suppose two iodines are attached. For the second one, two iodines are attached. For the third one, two iodines are attached. For the fourth one iodine is attached. Now, when coupling occurs, what happens? All the four iodine atom go to one side, suppose in one side, and here also all the three go to one side. So there is coupling between two adjacent tyrosine molecule. So what will happen finally in the chain? Finally, in the chain, there are some cases where four iodine atoms are there, some cases where three iodine atoms are there. Now, when there are four iodine atoms, we call it T4 molecule, and when there are three, we call it T3 molecule. Though till now they don't have any name or existence in the thyroglobulin. There are not separate existence here. They are present till now in the thyroglobulin molecule, but when they are released in the blood circulation, they have separate existence, and we will call it T3 and T4 molecule. We can call it this one as T2 molecule. We can call this one as T1 molecule. Okay. Sometimes students ask that what is why the naming of the thyroid hormone starting from T3, T4. So when one iodine is there, we can call it T1, or when two iodines, we can call it T2. Now two T2 molecule when fuses, T4 molecule is produced, and one T2 and one T1 molecule when fused or coupling occurs, then that is called T3 molecule. Now I am going to the previous slide again. So the coupling process occurs where? In the central hollow space. Now after the coupling process, then again there will be endocytosis process. So the thyroglobulin along with the T3 and T4 molecule, they are attached within this thyroglobulin, they are endocytosed. Now when there is need of release, there are some peptides. There are some peptides. Those peptides cuts the T4 at precise specific site, and finally T3 and T4 can be released. But they are not released always. When there is need, only they are released. After releasing, they, with the help of thyroid binding globulin, that is another protein, with the help of some albumin molecule, they are going to their target tissue. So, in the first part, we have learned that how T3, T4 molecules are produced. Thank you everybody. In the next part, we will learn how T3, T4 molecule actually acts in their target tissue. Thank you.